Bonjour tout le monde. Pour moi, c'est un plaisir de vous présenter Oscar Alejandro Escobar, Perez Escobar. Donc, il vient de la Colombie, je pense qu'il va parler de ça, mais il travaille à Kew Gardens à Londres. Et cette photo-là, c'est une photo intéressante parce que ça, c'était Bob Dressler, c'était l'orchidologue plus connu du monde à l'époque, qui travaillait surtout en Amérique latine. Et on a aussi Oscar, que c'est une personne qui a fait une belle carrière en orchidologie, en fait. Et maintenant, il travaille à Kew Gardens. Et j'ai rencontré Oscar quand il était étudiant de doc, quand il avait 22 ans. Donc, il est très jeune quand même. Donc, son premier article, c'était un article dans le journal Systematic Biology. Et ça fait longtemps de ça. Mais après ça, il a continué à faire la recherche sur beaucoup de choses, et surtout les orchidées, c'est sûr, et aussi la biologie tropicale. Et on voit surtout dans la partie des montagnes d'Amérique du Sud. Et aussi, c'est un papier très récent et très téléchargé, cité quand même. Et de plus, d'ailleurs, il s'intéresse aussi pour l'archéogénomique, donc il va nous présenter ça aujourd'hui, et la conservation d'écosystèmes tropicaux. On voit ici une lettre qu'il écrit en science sur les menaces aux mines en Colombie, en fait. Donc, pour moi, c'était un plaisir de vous présenter Oscar et merci beaucoup pour accepter l'invitation. If you don't understand anything, no worries, my French is...
C'est parfait, hein, on dirait, hein, c'est très bon quand même. Hein. Et alors, j'aimerais bien partager mon écran. Mais en fait, j'aimerais bien dire de grand merci à Juan Carlos pour l'invitation. Et Juan Carlos était un grand collègue pour moi, une grande inspiration quand j'ai fait mon doctorat à ma thèse en Munich. Ça fait déjà longtemps. Et Juan Carlos m'a donné beaucoup de bons conseils qui sont alors très utiles aujourd'hui. Et oui, merci pour l'invitation. Il fait que je parle maintenant en anglais parce que mon français, ce n'est pas très bon assez pour faire la présentation en français. So, thank you very much for inviting me to talk today. It is a great pleasure and yeah, it makes me very happy to see Juan Carlos again and doing so well. And thank you for the introduction. Today, I'd like to talk to you about a project we have been working on for already, 4 years? It's still ongoing, but what I want to present to you today is one of the outcomes of this project, of this ongoing project, which is about the evolution of date palm as investigated through ancient DNA and molecular clocks. And so this presentation will be divided in two parts, two segments. One where I will talk a little bit about what I'm doing in Kew Gardens. And this is because I am quite interested in establishing new cooperations, expanding my network. And then the second part would be devoted to the project on date palms themselves. Can everyone hear me and see my screen as well?
Yes.
Okay. Yes.
Cool. And if you have any question at any point, just feel free to ask. I don't usually like to wait towards the end because one tends to forget the questions. But anyways, feel free to interrupt. As Juan Carlos mentioned, I'm a Colombian scientist. I have been based in Kew Gardens for already 6 years. I came here as a postdoctoral fellow after I finished my PhD in Munich. And here in Kew Gardens, since almost 3 years already, I lead a small team of researchers. Basically, one postdoctoral fellow, that's Dr. Ai-Chun Hu, one postdoctoral research assistant, that's Dr. Natalia Shiomska, and a brand new PhD student, Charlotte Phillips. And I would like to warn you that, as Juan Carlos said, I am not a palm person. My main emphasis of my research is basically orchids, different aspects. But then I like to learn about different things, work in different modes, because this helps me learning different techniques as well, as you will see later. And so most of my research as a Latin scientist is focused in the American tropics. And I could say that most of my research revolves around everything that has to do with the origin and maintenance of plant diversity. Basically, I try to understand why some areas in the American tropics are so devoted or devoid of diversity, whereas some others have so much diversity. And then to answer these questions, I rely on phylogenetic, or let's say phylogenomic frameworks and trait data. And one example is, for example, these papers that Juan Carlos was mentioning before, where here in this figure on the right, we built the first, I could say the first framework at the genus level of orchids using target capture. So these are basically hundreds of supposedly low copy nuclear genes, right? And once we have these solid frameworks, I apply phylogenetic comparative methods to understand, for example, how biotic and abiotic variables have influenced the evolution, or let's say the diversification of orchids across long time scales. But then more recently, and this given the controversy that, for example, microevolutionary approaches have attracted, basically that is, not so reliable. They are not so reliable to use. I'm shifting a bit towards microevolution, so trying to compile data, high throughput sequencing data for individuals, and with the idea of conducting population genetics and understanding speciation and so on. But because these data sets are very scarce in orchids, since we have very few model organisms, when one thinks about how to learn new techniques related to population genetics, genomics, etc., one great idea is crops, of course, because there's a lot of money being invested on them. There are hundreds, if not thousands, of individual sequence available, like free to download in NCBI. And since a couple of years, in collaboration with our colleagues, including Susanne Renner and Guillaume Chomicki, two very inspiring scientists, on my side time, I've been learning techniques on population genetics and genomics. And more recently, let's say thanks to fate and again to Susanne, I have been discovering, let's say, what ancient DNA is and paleogenomics, something that you might have heard recently quite a lot, given the Nobel Prize that was awarded to Svante Pääbo, I think, who is a paleontologist also working in ancient DNA, right? And today I would like to talk to you about a project I have been working on date palms and ancient DNA. Then you wonder why palms and ancient DNA, right? Well, let's say more palms. And one reason why I decided to start working in palms is that here in Kew Gardens, we have the expertise on palms. We have John Dransfield and Bill Baker, who know really a lot about this taxonomy and systematics of palms. Also because orchids grow nicely on palms. But also because orchids are they're like what we call cornerstone species, basically species from which humankind depend a lot, and not only humankind but also many animals, right? Like birds, mammals, etc., etc. And for humans, they represent millions of dollars in several crops. And then of all the crops, palm crops like coconut, I think that, in my opinion, the most interesting one are the palms, because they are one of the earliest domesticated tree crops, right? But also because they have a fascinating and well-recorded history. And this is thanks to this is reflected on the many, let's say, objects that we find across history and different civilizations. So there's, for example, here coin engravings. They date from thousands of years to hundreds of years, which explain or kind of reflect or give the idea that date palms have been very important for us. Oh, we have, for example, these stone panels here from Iraq. And the reason why date palms have been so important for us is because they played a very important role on the domestication of other crops. As in, think of this, the habitats where date palm grows, oases, they provide an ideal environment for many other crops to grow, to flourish, right? And not only the importance of date palm is reflected on, let's say, artifacts like coins and panels, but also the utility for humankind as a food source and material source is reflected on the many, let's say, fragments of plants that we find through history. So here, for example, we have char seeds, date palm seeds. Sorry, phone is messing around. And because of the date palm represents so much for us as humankind, it has been thoroughly studied. There are like 2 genomes of reference, which are quite good. And as I said it before, there are hundreds of individuals that have been sequenced in great detail that are available to basically play around in NCBI. Right, so the date palm is known as Phoenix dactylifera and belongs to the genus Phoenix, which includes around 14 species. The species boundaries in the genus are not so clear, so sorry, qué pena que estoy en una charla, estoy en una conferencia. Claro, sorry, just give me una presentación sobre palmas.
Lo recogí mañana. Si llego hasta
Vale, sí, señor. Sorry, this was Eric Taxater. He's like the world authority on a genus called Epidendrum. So as I was saying, Phoenix dactylifera belongs to the genus Phoenix, which has 14 species, but no one has really tested the species boundaries in the genus. And Phoenix is distributed in Africa, Asia, and the Mediterranean. And while we don't have, so think of this, while Phoenix dactylifera represents millions of dollars for the economy, we don't have yet a solid phylogeny of the genus, right? But let's say that most of the species that have been studied so far and sequenced, when they are put into a phylogenetic context, most of the topologies that you see come like this. So the image that I'm highlighting here, where Phoenix dactylifera forms a grade, a non-monophyletic group with something that we call Phoenix atlantica that is distributed here on Cape Verde. We know that it's in fact not like a real species. Could be the same gene pool, represent the same gene pool as Phoenix dactylifera. But one key factor here is that Phoenix dactylifera in most of the phylogenies come as sister to Phoenix sylvestris. So we could say that the closest wild-living relative of Phoenix dactylifera, the date palm, is Phoenix sylvestris, something that grows somewhere here. And what you see here in blue is a distribution of Phoenix dactylifera. We know that the date palm was first domesticated in the Arabian Peninsula, somewhere like 6,000 years ago. And then there was a subsequent introduction into North Africa, right? But we don't have many, let's say that the archaeological records supporting this are not like really that abundant so far. Is everything clear so far?
Yes.
Okay, cool. Great. Something important I forgot to mention. We have some idea that, and one of the reasons why there is no like a solid phylogeny of Phoenix, apart from the fact that there is like a lack of sampling, so that we have a lot of evidence that there has been a lot of gene flow from and to Phoenix dactylifera and the closest wild-living relatives. So that's something that we have to keep in mind. And so what I'm presenting today has been, by the way, inspired a lot on the work of some collaborators, mostly Dr. Jonathan Flowers, Professor Michael Purganam, who is a tremendous figure on plant domestication, you might have heard of, and Muriel Gros-Balthazat, who is now an independent researcher in France, in Montpellier. And one of the coolest things that they found on their research is that they found a strong signature of gene flow between Phoenix dactylifera and something that we call Phoenix theophrasti. So if we go to the previous slide, this phylogeny, we can see here Phoenix theophrasti, which is not the immediate sister to Phoenix dactylifera, right? And basically what this paper found is that there was some major contribution, like gene flow contribution from Phoenix theophrasti to dactylifera. And they were actually able to locate which parts of the genome of Phoenix dactylifera were mostly affected by that introgression, like the signature of the introgression, basically. But something that wasn't really answered is the timing of these interactions. And in fact, where there was further evidence supporting gene flow between date palm and something else than Phoenix Theophrasti, right? And this leads us to the goals of this first part of the project, right? Which was basically to try to sort out the different genetic contributions of Phoenix species towards the date palm we consume and the timing of such exchanges. And so to answer these questions, what we did is that we basically sampled a bunch of accessions from NCBI, 39 modern accessions. So those are basically more individuals, cultivars, representing 4 species, and including obviously the date palm Phoenix dactylifera. And then from these 39 accessions, and using one of the most recent genomes of reference that is actually fragmented into 18 pseudomolecules. It's not a complete genome, but I think that these 18 pseudomolecules represent 50% of the nuclear genome. What we did is that we mapped these reads against this nuclear genome, and we inferred something called genotype likelihoods. And then from this kind of data, we inferred a mixture and integration tests. Something else that we drew to the analysis is a sample that we found in our collection that is dated to be 2,000 years old that I will introduce later. And then in the end, what we did with this data set is to produce a phylogen that we calibrated in order to try to understand where there were evidence of gene flow, like in, let's say, ancient times, like going backwards 100 to 200 million years. And the item that we used in this research that has 2,000 years actually comes from our collection, which is curated by Professor Mark Nesbitt. Our economic botany collection holds about 500 items that mostly come from Egypt and South America, a few from South America. And among these 500 items, 10 of them belong to date palms, and you can see here some examples. These are beautiful things to see, and if one day you come and visit the gardens, I will be very happy to show you the collection. This is like really fascinating. It's like a time trip whenever you go and see these items. And these materials, they represent at times, they represent a time scale, they cover a time scale between 3,500 years and 2,100 years. We have to be a bit careful with this information because not all these items are radiocarbon dated. So sometimes the age of these items are estimated based on, for example, the soil strata where the item was found. And this can be a bit ambiguous, right? So in order to be sure, so what you are seeing here is the item that we included in our research. And something that I forgot to mention, sorry, is that one reason why we decided to rely on ancient DNA, or let's say items that have thousands of years, is because they obviously represent like a timestamp of events that might have happened that sometimes we cannot see when we rely only on modern collections. This is because as like evolution goes through, right, some genotypes might get lost, and some of these genotypes might, for example, carry the signal of integration, hybridization, whatnot, that might get lost because of selection, right? And so the item that we selected for this research, you see it here, this object has a funny story, and it's that when it was first selected by Mark Nesbitt from the collection, he thought that it was something that he called a headpad, basically an item that ancient Egyptian civilizations were used to rest their heads. And actually, as we were writing the manuscript, that was the description of this item that we included in the text, a headpad. But then Mark wasn't like really convinced, and then he decided to contact some colleagues in the British Museum, and we were very lucky to find out another item that basically matches very much the morphology of ours, but that was much better documented. And as it happens, the real, let's say, utility and identity of this item is nothing related to a headpad, but something called a jar stopper. It's basically a device that people at the time used to use to seal jars that will contain beer, wine, food, etc. And then that was going to be later on covered in clay. So this item comes from a place called Saqqara, animal necropolis of Saqqara, which is very famous. I will talk about it in a minute. And what you're seeing here in this plot is the validation of the age of the item. which is around 2,100 years old, plus minus 23 years, something like that. Oh, sorry. And this is very important, as I said, because sometimes some research based on items that are supposedly thousands of years, they have a huge ambiguity age, and this is because they haven't like properly radiocarbon dated, right? So this is the place where the item was found. So Saqqara is a UNESCO site, and is mostly known from a pyramid, the pyramid of Djoser, which was built throughout the Old Kingdom, and also because millions of mummified animals have been found there, which has made the news. And Saqqara is somewhere like 20 kilometers south of El Cairo, I think. I think it's somewhere here in the map, if I remember correctly. Here, Saqqara. Right, so now moving up to the results. Once we sequence this item, as I was discussing earlier with Juan Carlos, this is not a really easy task because as, let's say, one collects plant material, right? The plant material starts to degrade, obviously, the DNA starts to damage, like happens to our DNA as well. The fact is that when we are alive and when plants are alive, we have a repairing mechanism that avoids mutations. Once the DNA breaks, avoid mutations to be permanent, to be fixed. But once you cut a piece of plant and starts to dry and so on, the DNA starts to be cut in pieces and, let's say, acquire what we call artificial mutations. And then conditions like humidity and so on will degrade further the DNA. You will get a lot of, let's say, fungi and bacteria feeding on the tissue. So often, let's say, the percentage of success of sequencing ancient DNA is very low, between 1 and 10%. And we were very, let's say, nervous about it. And this is actually exactly what happened in our case. Once we sent the sample to a specialized lab where basically you have to go with a gown and you have to disinfect like your gloves and everything, we produce around 30 million reads from this item that we call ancient Saqqara date pan. And what you see here in this slide is the outcome of the sequencing. And at first we were like very frustrated because of, let's say, 70% of these 30 million reads, which is not really much, were dimers. These are basically whenever you hybridize or you try to attach the adapters of your Illumina libraries, it's basically adapters hybridizing against themselves. And when that happens, and if you sequence a sample like that, most of the read data will go towards like this kind of thing, which is useless. It's basically not really like plant DNA, it's nothing. And of these then 30 million reads, 20%, something that we call exogenous DNA. This is basically real DNA, but belongs to something else that is not the pan or the organism that you are studying, right? It's again something that, while could be useful, is not of our interest. And finally, of these 30 million reads, only 3% was pan aDNA. And yeah, like 3% of our 30 million reads is not really much data. It's around 1 million Illumina reads, right? And of this small percentage, what we found out by mapping this data against the reference genome of DAPAN was that 60% of these reads matched the nuclear genome, almost 40% matched the plastic genome, and then 0.5% matched the mitochondrial genome. So in short, like sequencing archaeological remains can be very, very challenging. And then when you look at this outcome, something that you obviously wonder at first is, after you have invested so much money in this, is how much can you do with 1 million reads with such few data? And I hope that the next slides that I will show you will convince you that you can do many things. But before I go into that, I wanted to share with you something that we do to validate that this 3% of DNA is in fact real ancient DNA and not, let's say, modern plant DNA contaminations, which can happen, especially in a lab that manages so many samples from different plant groups, right? So what you're seeing here is a comparison between modern DNA and the ancient DNA. So the way this analysis is done, which is basically try to see patterns of damage and miscompositions or artificial mutations in the DNA, is that you map your read against a reference, and then you try to calculate, to compute based on models, how many artificial transitions you have or transversions, right? So usually in ancient DNA, the most common misincorporations or artificial mutations you see are C2Ts, and they tend to occur towards the end or the beginning of the DNA fragments. And that's exactly what we found. So this plot here on top is modern DNA. Basically, one NCBI accession reads data mapped against a plastic genome. And what you see is that basically there are almost no misincorporations towards the end of the fragments, where this is like read position, right? And this is the beginning of the read. And this plot that you see here is cumulative frequencies of C2Ts misincorporations in the plus and minus strand. Another thing that characterizes ancient DNA is the distribution of the read fragments, where modern DNA tend to have much longer fragments, which you can see here in the plot. Whereas ancient DNA, which is here shown in the bottom, the DNA fragments tend to be overall much shorter, and tend to have this damage here towards the ends of the reads, the C2Ts misincorporations. So based on this analysis, we are sure that what we are working on, this 3% of reads, around 800,000 reads, is real ancient DNA from the palms, of course. And precisely because of this damage, this post-mortem damage, we have to be very careful that these artificial mutations won't affect all our inferences, right? So something that we do before we proceed to any downstream analysis, like phylogenomics or conduct a mixture or integration analysis, what we do is that we try to calculate, after removing these transitions, artificial mutations, is to see whether the data itself has a lot of what we call error rates, which in this plot here, in this case, is an excess of derived alleles in a given sample compared to another group. And also something that we call transition-transversion ratios, which usually in ancient DNA samples is quite high. And as you can see in these two plots, the transition and transversion ratios and excess of derived alleles is quite comparable to other normal accessions, not normal accessions, sorry, and modern accessions, which is what you see here and here. So these plots tell us that while there is some damage in our samples, it's not as bad as, when you don't account for this kind of bias towards the beginning of your reads. And this analysis, by the way, was done by my colleague, Dr. Natalia Shomska, who knows much more than myself on ancient DNA and population genetics. Right, so now coming back to the question of how much can you do with 800,000 reads, here is another. What you're seeing here is an analysis where we try to place into a phylogenomic framework our ancient DNA sample, which is shown here in pink. So on the left side, you see a phylogeny inferred from entire plastic genomes of modern accessions of different species of phoenix. So Theophrasti, Canarias, and Silvestris. This is Dactylifera, including Atlantica, which, as I said before, is not a real, let's say, not a good species. And what you see here on the right is a network showing the relationships inferred from nuclear data. And no, sorry, this is also plastoms as well. It's two different analyses telling us similar stories. We were not able to produce nuclear phylogenies here because we didn't get much data to infer meaningful relationships. And this is because if we try to include in an analysis only data that is shared between the ancient sample and all modern accessions, we get very few SNPs, mutations, to infer meaningful relationships. And something very interesting that we see here in this analysis is that we have all in blue here are accessions of Phoenix Dactylifera, the daypalm that we consume. We have here our ancient sample, which is put closely, or let's say most closely related to daypalms from North Africa. All of them that have asterisks are North African daypalms. The rest are Asian daypalms. And then in here, in Phoenix Dactylifera, we see nested Phoenix Silvestris, which we know is a good species. And while this pattern is not new, what this pattern can tell us is that there must be some kind of chloroplast capture or something that is driving this incongruence, right? Because in nuclear phylogenies, we know that Phoenix Silvestris is sister to daypalms, but here is nested. And by the way, here in this phylogeny, anything that has thick branches basically means bipartitions that have more than 90% of bootstrap support. So this is quite solid. Is everything so far clear?
Yes.
Then moving on to, let's say, the nuclear story of our ancient sample, we conducted ancestry, or what we call a mixture analysis on PCA, to try to understand how related our date palm is to modern accessions and see whether we were able to recover any trace of gene flow. And what we found here in this PCA, which was inferred from 35,000 filtered nuclear sites, this is nuclear data sets only, is that our date palm is more closely related to a mix of African and Asian date palms. And this basically validates that our date palm, our ancient acquired palm, is in fact a Phoenix dactylifera, right? Belongs to the gene pool. And what you see here, these two pink dots is Phoenix atlantica, which again is another date palm, right? We have here in this corner Phoenix theophrasti, by the way, which is something we knew there was gene flow with already. But what is most interesting is that in the mixture analysis, when you consider 3 or 4 ancient populations, which is what the model told us is the best, you see that here the date palm, the ancient acquired date palm, which is these bars here. When you assume 3 or 4 populations, at 3 populations, part of the genome can be traced, a very small part of the genome can be traced to Phoenix theophrasti, which are this set of individuals here, 4 individuals. And when you assume 4 populations, you see that trace of a mixture disappears, and then one small portion, let's say 10% or less than 10% of the genome of this acquired date palm can be traced to Phoenix dactylifera, but Asian populations, which are shown here, these are all Asian populations and these are African. And these results are very similar to analysis conducted without ancient DNA on North African populations. So we were able to recover the same signal of integration between Phoenix theophrasti and North African date palms in our ancient date palm. And most importantly, most of the genome of the acquired date palm belongs or can be traced to North African date palms, which are these bars here. Right, but then this is not really evidence. The fact that you can trace part of the ancestry of a genome from one individual to other populations is not like a solid evidence for a gene flow. So in order to, let's say, find a more conclusive answer and actually test whether there is evidence of gene flow between our ancient sample and other species of Phoenix, we conducted something that is called the ABA-BABA test, or also known as DSTATS. Is something that I'm still like trying to do my mind and understand really well. It's something super complex. But in short, it's a non-parametric test where you try to see, depending on which position your samples sit, whether a set of samples share derived or ancestral alleles, depending on the position where these samples sit. And in short, what we found is that we recovered the same signal of integration that our colleagues, Jonathan Flowers, Muriel Baltasar, and Michael Burgana, which is this plot here shown, where our ancient sample here shown in position H1 was sharing more derived alleles with Phoenix theophrasti than Phoenix sylvestris, which is the immediate sister, right? But surprisingly, we also found nuclear evidence, and this is again all done in nuclear datasets, that our date palm, this acquired date palm, shared also derived alleles with Phoenix theophrasti, knowing that Saquara date palm is more closely related to Phoenix atlantica, which is again a North African date palm, right? And this is really cool because the same results were confirmed on a study that was published 2 months before our study by the same team, Muriel, Jonathan, and others, where they basically germinated seeds of date palms that they were found to be 2,000 years old, and I think they come from somewhere in Israel. And they found the same signatures of integration that occurred at that time. So in short, what this analysis is telling us is that the date palm, the North African date palm, shared genes or exchanged genes with Phoenix theophrasti and Phoenix sylvestris already by 2,000 years. And this is as inferred from a sample that has that age, right? Something that it would be difficult for us to inform or to learn if we don't have ancient DNA or ancient material. But then this doesn't really tell us whether other events of gene flow that go well beyond, let's say, pre-human times happened, which can occur, right? So in order to investigate that, what we did is that we infer a phylogeny of a set of species of phoenix that you can see here, where we have date palm, and we divided or we differentiated between Asian and North African populations. We also included individuals of Phoenix sylvestri, Theophrasti, and then we conducted analysis, dating analysis, and this was done by my colleague and wife, Sidonie Velor, and Maria Fernanda Torres. And these analyses were conducted in VIS2. We use molecular clocks, relaxed molecular clocks, and we use something called SNAP, where you basically can feed in information on populations. Like you are not dating a phylogeny using a single individual per species, but you said you use a set of individuals per species, and you can define populations. Something that we found that was very striking is that when, let's say, you take the entire genome of Phoenix dactylifera and infer phylogenies from every window, which is what we did, is that the most dominant topology is this, which we call the species tree, or the true relationships, right? Where Phoenix sylvestris is sister to the date palm we consume. But when you look into other, let's say, conflicting topologies that are also dominant, but not as dominant as the species tree, you see here, for example, that then Phoenix theophrasti comes as sister to the date palm we consume and puts its most recent common ancestor diverging around 5 million years plus some minus 7 years, which kind of gives us an idea that this gene flow, that there was other gene flow events that might have predated, human times. And then another dominant topology that you get is the one that supports, let's say, more recent gene flow events, like the ones that were recovered by Flowers and colleagues, where Phoenix theophrasti is placed as sister to North African date palms, right? And this was confirmed when you try to assess which are the most dominant topologies across chromosomes. You find that the species tree, which is again this blue topology you see here, is most dominant across all the chromosomes of Phoenix and dactylifera. Then you have the second topology here, which is the one shown in pink, which is the same gene flow pattern recovered by Flowers. And then on a smaller minority, you find this third topology, which is the one that supports an even more ancient gene flow event between Phoenix theophrasti and dactylifera. And so this analysis also tells us that these different topologies are not derived from something called ILS, incomplete linear sorting, something that you might have heard. And the evidence for that is that often whenever you have incomplete linear sorting, you will see that there is, not always, but frequently, there is one main topology, which is the species tree. But then under an ILS scenario, you will find that all other topologies, conflicting topologies, are present in your genome or your genes. They might tend to occur in equal frequencies, which is not the case here, right? These, for example, 9.13% represent many other possible topologies, actually 7 topologies, which actually occur in equal frequencies. So these other 7 topologies, that incongruence might be product of ILS, right? But that requires more investigation. And this analysis was conducted by my colleagues Sidonie and Ben Kunhoise, who is now a postdoctoral fellow here in CUBE. Right, this brings me to the end of my presentation. I hope I convince you that working with ancient DNA is really exciting, but it's also challenging. But once you manage to crack on like a proper protocol, you can learn like really cool things. I hope that I also convince you that our ancient date palm, is something that is very closely related to dactylifera and Atlantica, which in the end are the same species. But then when you conduct proper tests of integration, we found the strong evidence of gene flow between the date palm we consume, Phoenix theophrasti, and Phoenix sylvestris. So just think that there have been a gene exchange between these 3 species from 2,000 years old back to 5-6 million years. And that's it. Thank you very much for your attention. This work wouldn't have been possible with all my colleagues, which are knowledge here in both. And I'm very happy to take any questions.
Thank you very much, Oscar. Very nice presentation.